So today I have Dr. Kristin Sainani and Dr. Regina Nuzzo with me. Kristin Sainani is a professor of epidemiology at Stanford University. She teaches courses in statistics, clinical research, and writing, provides statistical support for clinical studies, and writes about medicine, science, and statistics for a range of audiences. She authored the health column Body News for Allure magazine for a decade. She's also the statistical editor for the journal Physical Medicine and Rehabilitation, and she co-authors a statistics column, Statistically Speaking, for this journal. She teaches the popular massive open online course, Writing in the Sciences, on Coursera. She was a recipient of the 2018 Biosciences Award for Excellence in Graduate Teaching at Stanford University. And Regina Nuzzo is a professor of mathematics at Gallaudet University and lecturer in population health at Stanford University. She teaches courses in statistics, science writing, and data communication for students, clinicians, researchers, and other working professionals. As a freelance journalist, she's covered topics in health, medicine, and research, including preclinical and clinical research, clinical trial design, and research rigor and reproducibility for publications such as New York Times, Los Angeles Times, Cancer Today, Scientific American, and Nature. She received the 2014 Excellence in Statistics Reporting Award from the American Statistical Association, and she gives workshops, short courses, and keynote talks about statistics, research, and communication to audiences worldwide. So thank you, Kristin and Regina, for being with us today, and I will go ahead and pass it on to them for their presentation.
Thanks, Jackie. So today in this webinar, we're actually going to discuss 4 case studies that have important lessons for the design and analysis of clinical trials. And they actually have also some pretty interesting interventions, things like wine and hookworms and some digital health products. So Regina, this first case study is super interesting because they actually randomly assign people to drink wine or mineral water every day for 2 years.
I'm wondering how I can sign up for this. Is it too late?
The study came out in 2015. It did get some headlines in the media, as you can imagine. Here's one of them here. It was specifically in people with type 2 diabetes, and they did claim to find some health benefits for wine in terms of cholesterol, lipids, and insulin sensitivity.
Which I think is good for our happy hour tonight, right?
I totally want wine to be good for your health, absolutely. But hold that thought because we are going to scrutinize those findings in a minute here. I do want to give the authors a lot of credit, though, because as you can imagine, it is not easy to do a randomized trial of alcohol drinking.
You know, I think this is super important, Kristin, because I see these types of headlines all the time, and they're observational studies. And of course, you can't get causation from observational studies because here's where you're observing people who have already chosen to drink wine or not. So if I'm the sort of person who likes a nice glass of red wine at the end of the day, I am probably different in other ways that also affect my health.
Exactly right. I mean, they've actually found that wine drinkers tend to be more educated. They tend to have more money than non-drinkers. So how do you know it's the wine per se versus these other factors that's influencing their health? That's why you need randomized trials.
Of course.
So let's actually dig into the study behind the headlines. This was called the Cascade trial. It was a phase 3 parallel randomized controlled trial. So phase 3 means they were looking at the efficacy of wine. Parallel means everybody was assigned to one group and you stayed in that group the entire 2 years of the trial. The participants were 224 adults with type 2 diabetes. They were all non-drinkers to start because they wanted to see the effect of adding wine to the diet. The interventions were 5 ounces daily of red wine or 5 ounces daily of white wine for 2 years, or you could have been put in the control group, which was 5 ounces daily of mineral water. The study did provide little bottles of water and wine for the participants. It was randomized, so you didn't get to choose which group you were put in. However, it was not blinded.
It's kind of hard to do that.
Yeah, you can usually tell if you've had a glass of wine, right? So hard to blind. For the purposes of this discussion, though, I'm going to focus on the outcomes of this study. So I went to the report, which was published in the Annals of Internal Medicine, and I extracted all of the outcomes that the authors reported. These were the 2-year changes since baseline, and they labeled 5 of their variables as primary outcomes. These had to do with cholesterol, lipids, and insulin sensitivity. and they labeled 10 additional variables as secondary outcomes. These were other measures of insulin sensitivity, cholesterol, lipids, and things like blood pressure and weight.
Right. So, Kristin, I'm seeing 5 then primary outcomes. That is a lot. Normally, we like to see just 1 or 2, right? Because every time you test a new outcome, you're opening yourself up to another risk of getting a false positive there. So do they control for that or adjust for that in their analyses?
No, they did not do any adjustments to the p-values for multiple testing. And the worry is if you have 5 primary outcomes, that's going to greatly inflate your type 1 error if you don't adjust the p-values, which they did not. Yeah. It gets a little worse than this, though. I want to give the authors a lot of credit, though, because they did pre-register their study in clinicaltrials.gov. So pre-registration means that you publicly release your protocol before you even start the study. And pre-registration is great because it kind of locks you into the protocol. It helps researchers to avoid the temptation later of just changing everything in their protocol after they see the data. So super important to pre-register. That means, though, that their protocol is available in clinicaltrials.gov. Anybody can go and check, did they actually stick to their protocol? So I went ahead and did that. Specifically, I looked at the outcomes that they had in clinicaltrials.gov. So in clinicaltrials.gov, they said that their primary outcome measure was going to be a 6-month variable, not a 2-year, and only 1 variable, glycemic control, which is actually a little vague. I think that might be hemoglobin A1C, which was not one of the primary outcomes they had in their paper. And they only listed 1 secondary outcome, which was CVD status, which I think is cardiovascular disease. You either have it or you don't, a binary variable.
So no matter what, this does not even match anything at all what you put on the previous slide, right? This is completely different. They didn't
this is, let me show you what that was actually in the published paper. Totally a mismatch, right? None of these are the same. So this is actually a practice that we call outcome switching, and it's not good. If you are allowed to choose your outcomes after you see the data, well, that's going to greatly increase your chance of what we call a type 1 error, a false positive error.
Not good.
Not good. And actually, let's go a little further and do a little thought experiment here, Regina. So we can imagine if wine does absolutely nothing, if it has absolutely no health benefits, we would actually still expect to see some statistically significant effects in a study designed in this way. And we can predict exactly how many. So we need to know that in the Cascade trial, they used a 5% significance level, meaning they would declare results statistically significant if the p-value is less than 0.05. That's very standard, but it creates a 5% type 1 error rate. And I'm counting in the published paper, there were at least 120 hypothesis tests that they ran. So remember, 5 primary, 10 secondary outcomes. That's at least 10 variables they looked at. I'm sure they even looked at things that weren't in the paper, but at least 15. And remember, they had to do 2 comparisons for every outcome, red wine versus water, and then separately, white wine versus water. And they measured everything at 4 time points. And they analyzed those time points separately rather than putting them together in a single repeated measures model. So that's a lot of hypothesis tests.
Okay, so to answer your little pop quiz for me, how many significant results would we expect? First of all, you got that 120 from multiplying 15 times 2 times 4, and 120 is a lot of hypothesis tests. And you said that the methods would allow for 5% of them to be false positives. So 5% of 120 is 6. So, right, 6 false positives. There we go.
Right, if wine does absolutely nothing, we're still expecting to see 6 statistically significant results. And this is this idea that if you torture the data long enough, they will confess to anything.
I love that quote. You know, but I think sometimes people get maybe a little confused about how does torturing data, right, lead to false positives. It sounds a little strange. And I would like to think of the role of random chance in all of these measures. It plays a big role, and like cholesterol level. So every time I go into the doctor, my cholesterol changes just a little bit. So assume one does nothing, but at one of these time points, like 18 months, You just happen to have in the red wine group more people that have lower cholesterol levels. So wine does nothing, but it's going to look like it does something. And so that's going to come up statistically significant as a false positive. And the whole torturing thing is like, you know, there's going to be a false positive somewhere. So you turn over all the rocks, right? As many as you can until, aha, you find 6 false positives maybe.
Exactly. That's exactly right. And Regina, you want to take a wild stab at how many statistically significant results were in the Cascade paper?
I am hoping it's not 6. Please tell me there's not.
It was actually 5. And do you want to guess which 5 variables specifically were statistically significant?
There were 5 primary variables, so I am sadly assuming it was the primary outcome.
It was exactly the primary outcomes. 3 came out significant for red wine, 2 for white wine, never for both wines at once. And all of the secondary outcomes were not statistically significant.
So, Kristin, does it look like to you, it looks to me like they collected a lot of information about a lot of outcomes, a lot of variables, good for them, and then analyzed everything they could get their hands on. And then after the fact, the ones that came up statistically significant, they put over in primary outcomes, they ignored what they said they were going to do in that pre-registration and changed everything after the fact. Yeah.
I mean, there's no way to know for sure what happened here, but it definitely looks like that's what happened. They found all the significant results and then selectively reported those as the primary outcomes. We actually call this practice p-hacking, where you're trying to find significant p-values and then selectively reporting them or manipulating your data to get significant p-values. So the problem here is that imagine if you're a reader of this paper, you're going to look at the paper and you're going to think the evidence is stronger than it is because you're going to assume that those primary outcomes were picked a priori. Now, we've looked carefully here and we know actually they weren't. So when I'm looking at these results in context and I'm saying, okay, 120 hypothesis tests, 5 of those came out significant, to me, this is completely consistent with chance. Like, in other words, I'm looking at that thinking, yep, this is what you'd see if wine does nothing. So to me, this provides no evidence that wine is beneficial. And I'm really sad about that because I absolutely I want wine to be good for my health, but there's nothing in here to convince me that this is what I'd expect to see if just chance is at work.
Yeah.
Take home points here. Randomized trials of things like wine drinking, super important, hard to do, but super important because you can't get causation out of observational studies. But the more statistical tests you run, the higher the chance of false positives. And this outcome switching, this is a form of p-hacking or cherry picking that can lead to false positive results.
Okay, Kristin, now this is the point, as we're going to do for all our conversations about these case studies, is rate the strength of evidence 1 to 5 stars, like we are wine connoisseurs ranking a fine wine. So what's your rating?
Yeah, Regina, I'm going to be able to give the evidence here only 1 star because, like I said, to me, this evidence is exactly what I'd expect to see if wine does nothing. So I'm not at all convinced that wine is helpful for health, sadly.
Yeah. So I agree with you, but I'm going to bump it up to 2 stars for me just because I love that they had the audacity to run a randomized trial of drinking alcohol, drinking wine. So 2 stars for me.
Fair enough.
All right, switching over now, I'm going to do case study 2. Kristin, get ready. You gave us drinking wine as a therapy for type 2 diabetes, and I offer you a maybe more palatable alternative, hookworms.
Hookworms, as in the parasitic intestinal infection hookworms?
On purpose, yes, in your body, intestinal worms in your body on purpose, yes. People were signing up for this as part of a clinical trial. And as you can imagine, the media enjoyed this. Pioneering trial. It's a study that will make many toes curl.
This is definitely making my toes curl, and I think I will stick with the wine. Thank you very much, and not go with the hookworms.
All I ask is that you wait until you see the evidence, all right? Open mind going into this. So I want to talk about the details of the study, but before I do that, I don't know about you, but I was really curious. Why are we even talking about hookworms for insulin resistance, right? What is going on there? So I dug into the intro section of the paper, and it was kind of fascinating. I wanted to share this. So did you know that for most of human history, they think that parasitic worm infections were pretty commonplace. And we just lived with it. And researchers have noticed that in areas where that is still the case, these types of metabolic, lifestyle diseases, type 2 diabetes and other inflammatory diseases, they tend to be lower. And then when public health workers have gone in and tried to lower the rates of the parasitic, infections, they have noticed that those metabolic disease rates have risen. Interesting.
So that's super interesting, but I'm going to look at that sort of with the same skeptical eye as we were talking about observational studies of wine drinking, because this is observational, and maybe the places that have a lot of parasitic worms, they also don't have a McDonald's on every corner, and then about the time people are coming in and eradicating those worms, maybe that's the time at which the McDonald's are springing up on every corner. So this could just be coincidental.
Right. So that is exactly, again, why we need randomized controlled trials, this time with very brave volunteers who are willing to do this. So let's get into some of the details. Like yours, this was a 2-year, 3-arm parallel trial. Yours was a phase 3, though, and this is phase 1B. And I think sometimes people may not realize when they see the headlines that the goal of a phase 1 trial is very different than the goal of a phase 3. It is not designed to test efficacy. It's not designed to see whether the therapy is actually working or effective. Sometimes they call these phase 1 trials first in human trials. And so the idea is just get a small group of brave people and see if they can even tolerate the therapy. Like, what doses can they tolerate? Do they have to drop out? What side effects do they get? Then they use that information to go on and design these phase 2 and then eventually phase 3 clinical trials where they can look at efficacy. So very different. I just wanted to make that clear. Chris and I also checked, of course, to see if the protocol was pre-registered. It was in the Australian version of clinicaltrials.gov, and they published their protocol in a peer-reviewed journal. And get this, they made their data publicly available.
So I love that. I love when authors make their data available, because as a reviewer, it's very helpful to me to have access to the data to be able to check things. It also gives me a lot of confidence in the research because the researchers were willing to share their data publicly. They don't have anything to hide.
I love the transparency. I feel like they get double bonus points for that one. So they looked at 40 adults who all were at risk for type 2 diabetes. Now, 40 sounds small, but it's not unusual for this sort of a phase 1 clinical trial. And they randomized them to one of 3 groups. You could either get a dose of 20 hookworm larvae, a dose of 40 hookworm larvae, or a dose of Tabasco sauce. And so the worms are microscopic, the larvae, but they put it on your arm underneath like an opaque bandage, so you couldn't see what was going on.
So the larvae are absorbed through your skin? That's gross. Don't eat them, they go through the skin. Yeah.
Are your toes curly now? Yes, in the first 6 hours or so, they do come in through your skin, and you get maybe a little bit of tingling, or burning sensation, maybe a little bit of a rash there. Then, in case you didn't know, the hookworms kind of make their way through your system and eventually arrive at your gut. And then they use their little hookworm heads to hook on to your intestinal wall. And that's where they lay eggs and make a new home for themselves, set up a house.
So that sounds gross, but I'm noticing that they have a really nice sham. The control condition is a sham intervention because that chili pepper sauce, that's going to make you think you got the worms. It's a good placebo.
Exactly. And they did tell them, right, oh, you may be feeling a little bit of a tingling. And I love this sham intervention. I think it is super important no matter what the intervention, but especially ones that have this sort of ick factor, right? So if you think that you were randomized into the arm that has hookworms, you might be changing your behavior, right? You might like lose your appetite, eat a little less, right? You might just feel sick to your stomach just thinking about it. And that's why it was important to make everyone think that they might have gotten that intervention, right? So then they can separate out what were participants' expectations or biases, or what were they expecting, unconscious or not, and separate out and really find out what was going on with the intervention. So I love that. The block randomization, that's nice for a small clinical trial because it helps ensure better balanced groups. And they did blind the participants, as we talked about, but also the researchers. So they help to control for those unconscious biases or expectations from the people that were treating the participants and that were collecting data from them. So they're nice. So here were the outcomes that they listed in their registered protocol. There were 3 of them that were looking at that tolerability and 4 of them that were looking at the efficacy after the fact.
So Regina, we just talked about in the Cascade trial, there's kind of a lot of outcomes, but actually this is very different because in a phase 1 trial, we're not worried about type 1 error because the point is not to evaluate the effectiveness of the treatment, it's to just look at safety and tolerance. So it's okay if you have more outcomes.
Exactly. It's different for that phase 1. I should say though, even with that in their secondary outcomes, they did control for multiple comparisons.
Wow, I'm impressed.
Yeah. Wow. I feel like they got some extra points for that one. Exactly. All right. So let's look at what the actual results were. There we go. All right. Serious adverse events, there were none. They were a little worried about anemia because apparently the worms can maybe eat your blood or something. No anemia in the participants, but there were some gastrointestinal side effects in there. A little under half in the hookworm group, none in the placebo, not unexpected. So this is when the worms are in your gut. So 3 of it's like loss of appetite, nausea. So 3 participants were not able to take it. They needed to drop out of the study, but the other 24 were fine. In fact, it went away. It was only in the first month and a half, and it went away without any medical intervention, without any other problems in there.
Regina, I'm noticing the other adverse events, the non-gastrointestinal, those are actually pretty similar between the hookworm and the placebo group, which kind of shows you why you need a placebo group because, people get headaches, things happen. You might feel, as you mentioned, you might feel icky if you think you have worms. So it's good to know that actually that's all really placebo effect.
Yeah, again, it was really good that they had a good sham intervention here. They actually call that nocebo effect, right? A placebo effect is you think you might have the intervention and it might be doing something good. The nocebo is you think you might have the intervention, you might have bad side effects. So they were able to separate that there, right? Okay, but mood. Let's look at the mood. The mood was actually slightly better in the hookworm group, Kristin, and this was during the pandemic.
Well, I guess they had a friend. They weren't alone during the pandemic. A little pet.
And picture you weren't alone during the lockdowns, right? You had someone to play cards with, watch TV. The completion rates were about similar between the 2 groups, so that was good. Now, again, they did not design this to look at efficacy, but while they were there, of course, they looked at it, and they found that the insulin sensitivity actually did improve in the low-dose group that works in the placebo. So that was impressive. And the weight, look at the weight. The average loss for people in the low dose group was about 5 kilograms at year 1, and it persisted on through year 2.
5 kilograms, that's over 10 pounds. So that's actually a meaningful amount of weight loss, Regina.
It is. They were very excited to see this, and I think that might help explain my last observation that all except for 1 person, everyone else in the hookworm groups said at the end of the study they wanted to keep their hookworms.
We're going to let them live there forever? Really?
Still not convinced, right? I'm going to convince you to come over, Team Hookworm. I think that this was great. So the researchers were very excited about this. Phase 1 trial, which means now they have information about tolerability. Not only can most people tolerate this therapy for 2 years, they can even maybe stand it longer than 2 years. They know what sorts of side effects to expect and what the course of that is. And they also got a little information about the dosing. For some reason, it looks like the low dose group did better than the high dose group, and they're not sure why. Maybe you just need 20 hookworm larvae and not 40, and that's it. So they're going to take all this information, then use it to design a phase 2 trial where they'll dig a little bit deeper into what's going on with the dosing, and then hopefully go on and design a phase 3 clinical trial where you can take a whole bunch of people and see how the hookworms perform in a big group. All right, take home points here. Phase 1 trials are not designed to look at efficacy. It's the phase 3 trials that look at efficacy. The phase 1 is really looking at tolerability, safety, those sorts of things. When you make your data publicly available and when you pre-register your protocol, you make people happy, you really increase your trustworthiness. And doing a good job of blinding with both the participants and the researchers in choosing a good sham intervention will help separate out any unconscious expectations or biases and let you really focus on what's going on in the intervention.
All right, Regina, how many stars are we giving that one?
You know, I love this. It was only a phase 1 trial, right? So they weren't looking for efficacy, but I think they knocked it out of the park for phase 1. For what they set out to do, they did a wonderful job. So 5 stars for me.
I have to agree with you. This was a really well done study, so I'm going to have to give it 5 stars. I am sorely tempted to take away a star for the X factor here, but I can't do that in good conscience because the evidence is excellent. Yeah.
I really admire your adherence to scientific principles.
All right, third case study, Regina, not quite as exciting as hookworms, but it involves a digital health product. And the idea here was to give people competition that might motivate them to engage in healthier behaviors.
I have to say I'm pretty competitive, so this might work for me.
Yep, me too. It just came out in 2023. It did get some headlines, and they focused actually on medical interns. So have you ever seen the show Grey's Anatomy, Regina?
Yeah, a bunch of beautiful young people, overworked, right?
Right. They're on call, they're sleep deprived. Somehow the characters on that show still have plenty of time for drama, but you know, usually medical interns don't get much sleep. So the researchers here, they wanted to help interns to get more sleep and more exercise. And so they put them on teams to compete, and they engaged them in competition through an app on their phone. So every week, the teams were randomly assigned to either be in a competition, either for exercise steps or for sleep. And they were randomized to face off against different teams, or they were randomized to be in no competition. So the intervention here is being in a competition.
Okay.
So they would get a notification to their phone, something like at 9 p.m. on Sunday, that would say something like, MGH surgery faces off against Northwestern Internal Medicine in this week's STEP competition. And then they'd get reminders during the week. Yale Psychiatry is leading in this week's sleep challenge with an average of 8 hours and 41 minutes.
That's way more sleep than I get. What are you talking about?
Right. I mean, yeah, I don't get that much sleep. I doubt interns do, but you know, this was the example in their paper, maybe meant for illustration of the notification. What the phone app actually looked like. But let's now dig into the study behind the headlines. So this was what we call a micro randomized controlled trial. And actually micro randomization, something we talk about in our course, Regina, super cool new technique that allows you to randomize people many, many, many different times during the study. The participants were in teams. Each team had to have at least 5 interns. There were 191 teams, and they were randomized weekly to either be in a competition for steps or sleep, or to not be in a competition. That's the control here. It was not blinded because you did know if you were in a competition or not. They did give a small monetary incentive. Medical interns don't make much, so you know, that can often up participant engagement. They had 2 primary outcomes. So for the during the step competition to no competition, they were interested in the average daily step count as measured by like a smartwatch, and the average step count for the entire team. For the sleep competition, it was the daily sleep minutes. The average daily sleep minutes was the outcome.
Mm-hmm.
Talk a little bit more about that micro randomization. It's super cool. So in a traditional clinical trial, you can think about something where maybe they're giving a drug and a placebo pill. It's actually very complicated. The logistics of figuring out the randomization, getting people the right pills, keeping everybody blinded, logistically very tricky. But if you have an intervention that can be administered on the phone, like, hey, you're in a competition this week, guess what? The phone can also do randomization. So that allows you to randomize people lots and lots of times during the study. I mean, week 1 of this study, all the teams were randomized either to step, sleep, or no competition. And then that was repeated in week 2. They were re-randomized. And then it was changed in week 3. And you could be in all of the arms at different times in the study.
This is what I love about micro-randomization, Kristin. So 50% of the time they were randomized to no competition. That means half of the time each team was serving as its own control, which is very statistically powerful. It's efficient. It's a very efficient use of sample size because especially if you have a lot of variability between teams. So like say the Stanford team is really good at sleeping, really good at walking around, then you want to be able to compare the effective competition on top of that, right? So it's really helpful when you have your own control in there.
Yeah, exactly. And just for fun, Regina, because I like math, there are 3 arms to this trial, and they were randomized for 12 weeks, so 12 different times. That creates 3 to the 12th, or 531,000 different possible randomization sequences. Clearly, you need to implement this on a phone and not with a person. All right, they did find a small but statistically significant effect when they were in the step competition versus no competition. They walked on average about 100 steps more per day.
That is walking to the vending machine, Kristin. I don't buy it.
If you were very sedentary, that could be a meaningful amount. And interestingly, when they dug into the data, they noticed something. They noticed that there was a significant interaction with time. And so the effect was bigger at the start. They were walking an average of maybe 200 steps more per day, and then it petered off as you went along. You might expect this with something like a digital health app, because, you know, bored of our apps on our phone after a while. They did not find any significant effect for either sleep or for their secondary outcome, which was mood. I was able to check, and I went to clinicaltrials.gov, and I was able to find the protocol, and I was able to check their outcomes. I noticed that it was exactly the same as what they published in their published paper in NPJ Digital Medicine, so that was great because there was no evidence here of outcome switching. Whew. But I did notice 2 red flags with this study. So when I went to clinicaltrials.gov, the study completion date was listed as June 30th, 2021. And unfortunately, the study took place in 2020, but unfortunately, they didn't actually post their protocol to clinicaltrials.gov until November 3rd, 2021.
So they didn't actually pre-register then. This is post-registration, not pre-registration, right? They're not getting their benefits, right?
Yeah, unfortunately, this is actually not pre-registration. So, you know, that's a red flag. And then the other thing I noticed that was worrisome to me, everywhere in the study, the protocol, the appendix, I thought this was a 12-week study when I first read it. In fact, in the methods section, they say we started on July 6, 2020 and ended on September 27, 2020. That's fine. But when I read way down deep in the fine print of the discussion section, I noticed this passage that worried me. It says, in fact, the data in our study has been collected for 26 weeks.
Whoa.
However, we noticed that the percentage of missing data increased notably as the study went on. Therefore, we chose to perform analysis on the first 12 weeks of the data.
So like you are probably going to say, I don't like this. So Kristin, it looks like this was actually a 6-month study, and people got maybe bored, or, you know, participation dropped off. So they decided to just only analyze data up to the 12th week and then get rid of 14 weeks of data. And why the 12th week? Why not 13th or 11th? You know, this is another cherry picking. This is another p hacking. You probably don't like it any more than I do.
Yeah, I mean, I don't know for sure what happened here. Again, it wasn't mentioned anywhere else, but to me, this sounds like, yeah, this was actually a 6-month study, not a 12-week study. And we can't just change it after the fact because that's hacking. And they could have been transparent here, right? You can imagine that they could have just said, hey, here's our 6 months of data. They wouldn't have found an overall significant effect for stepping, but they could have then said, but look, when we look carefully at the data, we noticed that there was a significant effect early on. There was an interaction with time. You know, it wouldn't have really even changed the take-home message that much to be transparent here, because small effect early on and it peters out because people get bored of the app. So too bad they weren't transparent.
Yeah.
Take-home points, micro randomization, super cool technique, but pre-registration means free before you enroll the first participant. And throwing out portions of the data, that's another form of p-hacking or cherry picking that can lead to false positives.
Yeah. All right, what do you say? What's our 5-star rating on this?
Yeah, so I'm going to give it 3 stars. I like the micro randomization, and I also think if they had been transparent, it wouldn't have completely changed the take home from the data. So, but I got to take some stars off for this dumping data.
Yeah, you know, I agree with you. For some reason, I want to be a little bit more critical here and bring it down to 2 stars, just because they weren't transparent and they could have been. And so that just I don't like that. But
very reasonable to me, Regina.
Right. Okay, last case study. I also chose a digital health product. This one was AI enhanced glasses for kids on the autism spectrum.
That sounds super cool.
Super cool. It's based on Google Glass. Do you remember this, like a decade ago, maybe?
So I remember there was like all this hype around Google Glass, and then I never heard about it again.
Well, that's because researchers are using it now. They realize it makes a good platform for developing other, products or therapies or interventions. This one in particular is to help kids with autism process and understand emotions on the faces of people around them. And it's kind of neat. So I want to take a moment to explain how it works, and just because I was very curious about it. So the kid is wearing the superpower glass, and it's connected to an app on their parent's phone. And you see in that corner, it has a little prism. That's like a display. That's where it'll flash things they can see out of the corner of their eye. And so they're interacting with their family members, and then the camera then will find a face and spot a face, and it will show up with a little green frame saying, hey, there's a face here. And then the cool part, the app then tries to categorize that face's emotion into one of 8 emotions, happy or sad or meh or something like that. Then it will translate that into an emoji that will show up for the kid. So the kid knows, okay, this is a happy face. And then you can play games with your family, guess the emotion, or tell some jokes and see if you can get a smile out of that.
That's super cool.
Yeah, very neat, very neat. So they did some exploratory studies, but I want to talk about what happened with their randomized controlled trial, first of all. So the study design, this was a crossover design with a waitlist control. And all that means is the families were randomized to either get the intervention first or the control phase first. And then after 6 weeks, they crossed over. So if they had the intervention first, then they went on to the control phase, they had the control phase, then they had the intervention. So every family was in both the intervention and the control, just in different orders, depending on what they were randomized into. This was registered at clinicaltrials.gov, but like yours, it was only after the study was completed, which
I'm noticing this a lot, Regina, with digital health interventions, that you go to clinicaltrials.gov and you find out actually it was registered, right before they went to publish the paper. So I think people aren't realizing that you have to register prior to enrolling any participants, not just when you go to publish the paper. So we need to get the word out about that.
Pre means pre. We got to start a little slogan there. The data was not publicly available, but they did say that they would make it available after the fact for researchers who would sign a release, that would contact them directly. So they worked with 71 kids, all with autism, that were already getting behavioral therapy during the week. And for the intervention, they asked the families to use a superpower glass for sessions of 20 minutes, 4 times a week over 6 weeks. And then the children still got their regular behavioral therapy. And then for the control, though, it was just the regular behavioral therapy, nothing else.
So Regina, that's kind of interesting because I'm thinking like the chili pepper sauce, that there was an opportunity here to do a better sham treatment, right? I mean, everybody could have gotten the glasses and then you could play a game that had nothing to do with emotions, right? So it's too bad that they didn't use a better sham control here.
I know. I feel like this is a missed opportunity because before we were talking about the ick factor, right, with the hookworms. Now I feel like maybe there's a coolness factor. So you're really excited because you're getting to play with this cool technology. I always get super excited. And that might change your behavior and change the sorts of things that you're noticing. So it's hard to control for those participant biases or expectations unconscious. They did randomize, and they blinded only some researchers, not the participants and not all the researchers. So this is what they had listed on the registered protocol. There were 7 outcomes, 4 primary, 3 secondary, and they were all measures of the kids' ability to socialize or, decipher emotions, this sort of thing. Some were done by blinded researchers, but some were done by parents.
But worry a little bit about the one done by parents, Regina, because I mean, the parents know if they got the glasses or not, so they could have the expectation that their kids would do better. So I'd worry a little bit about that. And then I'm noticing here that there are 4 primary outcomes. It's kind of like the Cascade trial. That's a lot. Did they adjust here for multiple testing?
They actually did adjust for multiple testing. Yes, they did bond for any correction. I know. Good for them. Good for them. Finally. All right, so let's see what the outcomes. First one, statistically significant. That was the VABS-2 socialization subscale. The next 5 were not. And then the BOSCC, it was mentioned in the registration, and then I couldn't find it in the actual published paper.
So not outcome switching, but like outcome losing, I guess.
The lost outcome. Yeah.
Yeah, I'm looking at this, Regina. I mean, yes, they got 1 significant, and I guess they did correct for multiple testing, so that's good. There's some control of type 1 error, but it's not terribly strong, the results.
Yeah, I'm not completely blown away by this. And the more I started reading, the more I found some potential problems, I think, that might have weakened the evidence here. And one we touched on a little bit. It was the crossover design. And so remember, the crossover design is you just get randomized 1 time to either the intervention and the control, and then you flip whatever phase you were in, you flipped. So with the micro-randomization, of course, you were randomized 12 times, right? So you had lots of potential things there. So these sorts of crossover designs are really nice, like with your competition app, because you get to serve as your own control. But guess what I spotted or thought of that was different than your app on this?
Right. So the competition, it makes a good one for a crossover design because you're hoping the competition works while you're in a competition. So you're not expecting any lasting effects. It's going to work during the competition week only. I think the glasses are a little different though, because I assume they wanted to train the kids in the long run how to recognize emotions. So if the glasses actually work and you get them first, you can imagine there might be some spillover into the control condition, like you're now a little better at recognizing emotions. So that's going to weaken the difference between the treatment and the control. It's going to weaken your effect size.
Yeah. I'm surprised they did this. I feel like this was a missed opportunity, and they might have been better with just a standard parallel trial where you randomize into one thing and stay there. Yeah. So placebo effect issues we touched on a bit. Even if they didn't have the glasses that play at other sorts of games there, they still could have asked the families, okay, get together and tell silly jokes for 4 times a week, for 20 minutes each, just to have the family time, control for family time in there. So the last one, I think might be potentially important. The families use the device on average only half as often as they were supposed to. So only an average of 2 times per week versus 4. And about 1 in 4 of the families dropped out because they found the device too challenging to use or too uncomfortable.
This is very problematic, Regina, because if the glasses do work, but they're not user-friendly, if nobody can use them, that's a problem. So why didn't they fix it and make it more user-friendly?
Right, especially because, as I said, they had done these exploratory studies before. And in one of them, they also had some a 21% non-compliance rate, so something similar. So they might have caught it earlier, right?
Right, they should have learned from that exploratory study. Why didn't they fix it after that?
Yeah, I know. So the problem is, and I double checked this, it looks like the researchers started this clinical trial while they were still recruiting for the exploratory study.
Okay, wait a minute. So exploratory studies, the whole point is to collect information to inform the design of the clinical trial, but they couldn't have done that here if they did them simultaneously. That doesn't make any sense.
Bingo, bingo. I think they really lost the benefits of doing that exploratory study. So they were iterating the product throughout the exploratory study and maybe throughout the clinical trial. But they really lost, again, a lost opportunity to take the information from that earlier phase study and then design something better there. Yeah. So I think overall these problems might have contributed to them getting some negative comments from other researchers in the press. And I found this quote particularly evocative. It said, "This is basically science's use as a marketing exercise."
Ouch.
Yeah. It's too bad, it really is too bad, because I feel like much of this was really preventable. So the take-home point here, you really need to not skip steps, right? If you're going to do your exploratory study, then make use of that before you go on to your later stage trials, and that will end up being stronger as a result. Really know your intervention and know the range of study designs that are possible, and then make sure you choose a study design that really fits your intervention, and do the same thing with your control comparison. Think about what might make a good sham intervention or a good control, so you can really get a clearer picture, maybe some stronger evidence out of this.
All right, Regina, so what are we going to give this one in terms of our star rating for evidence? What do you think?
I'm on the fence here. I'm going to give it 3 stars. I wasn't blown away by the evidence. It did some good things, but I feel like they had some missed opportunities that they could have done better. So I wish that they had presented some stronger evidence.
With 2 stars, I think, because I think there were so many weaknesses in the study design. The evidence was really not that strong. So 2 stars.
All right, so that wraps that up. Kristin, as always, thank you for all this scintillating conversation, hookworms and wine, and all kinds of good things. We turn it back over to Jackie now, and Jackie will take us. Yep.
Absolutely. So thank you, Kristin and Regina. That was an amazing presentation, really informative. We have a ton of Q&A questions coming in here. And thank you all for sharing your questions with us. We'll try to get to as many questions as we can. A ton of questions have come in, Kristin and Regina. So we'll start going through those here. So the first question that we have is related to case study number 1. So what if the primary outcomes were correlated based on previous studies?
Right. So you still need to be judicious in which one you pick. Now, the idea is if you had, if they were so correlated that they are pretty much the same measurement, right, then of course it would be like measuring one thing, but presumably cholesterol, insulin sensitivity, lipids are not super highly correlated. They may be somewhat correlated. So yes, if you thought they were all measuring the same thing, you would still pick one. And sometimes we do things like combine them into one measure, or we pick the one that we think is the most representative when they're highly correlated. There's ways to actually combine those outcomes into a single measure if you think they're going to be highly correlated.
Especially for the primary outcomes. Yep.
Yes. We're most worried about the primary outcomes and keeping the type 1 error rate low on those.
Yep.
Okay, also related to case study 1, what would have been a better statistical test to prevent false positives with multiple testing?
Right. So if they were really set on having 5 primary outcomes, of course, we believe that actually those outcomes were chosen after the fact. So both of those pieces are a problem, not only having 5, but choosing them after the fact. But let's say that in the design of the study, they had, for some reason, really wanted to have 5 separate outcomes. Then in the design of the study, they should have also built in some kind of control for multiple testing. Bonferroni is the most common. There were actually some more sophisticated ones that they might have chosen there, but they would have had to have done that all up front, not after seeing the data.
Okay. And so I think that kind of partially answers our next question, but if you have any additional thoughts on what are some of the key steps that we need to follow to identify the statistical false positives in any research article instead of kind of blindly believing it.
Yeah, so I mean, you never know which ones are the false positives or not. You can't know that. If we knew that, we wouldn't have to do the study. But the key is always looking at the totality of the evidence. So authors, consciously or unconsciously, are going to try to draw your eye to the few positive results, and you need to step back and say, let me look at the totality of the evidence. So sort of what I was doing is counting up the number of hypothesis tests and saying, the context here is we've got at least 120. So you have to use judgment and just not overly focusing on the significant ones and saying, well, but what was the background? How many things did they test?
Okay. Anything to add there, Regina?
Nope. Kristin's got it.
And then also regarding the false negatives or false positives, how do you mitigate this issue when you're conducting multiple statistical tests on a set of data? And then will the adjustments also increase the risk of false negatives?
Right, good question. So, so the first part, there's a lot of design choices you have. So you can choose to have only one primary outcome, and then you don't have to adjust your p-values. Or again, if there's some pressing reason you want to have more than one, then you can use one of these adjustments for multiple testing. So there's a lot of decisions you can make in the design phase to try to mitigate the type 1 error rate. Yeah, if you use these tests for the multiple testing, what you're essentially doing is making the bar harder, right? Instead of needing a p of 0.05, maybe you need a p of 0.01. So that absolutely will increase your false negatives. There's always a trade-off between false negatives and false positives.
Okay. And then the next question is, so is this concept of cherry picking widely known about in the clinical industry? And they're just curious how common it is for clinical trial results to be misinterpreted.
No and yes. Go ahead, Kristin. You can take that one.
I'm going to let you take that one, Regina.
Okay. It's hard for us to say what is widely known, because of course there are pockets of people who are doing really great research, right? Who know this, and they're doing rigorous research, and they're labbed, and they're churning out great researchers. But there are a significant number of people out there who don't know. Either they're running the trials themselves, or they're analyzing the data, or they're interpreting the results, or they're setting policy or making decisions from this. And well, Kristin and I addressed, some of that in the course on what to look for and how to avoid that. So cherry picking is definitely one of the big ones, not just in clinical trials, but in science in general. Yes. What was the second half of the question?
The second half of the question was how common it is that clinical trial results are misinterpreted.
Ooh. Again, hard to say, because I haven't done an overview of a survey, right? And the conservative statistician in me doesn't want to go out on a limb and say something. But I think it's fair to say, based on Kristin and my reading of the literature in the media coverage and the press releases it gets, that there are a lot of misinterpretations.
A lot of overhyping.
Yep.
Okay. And then regarding case study number 2, there was a question about explaining the term block randomization.
Oh, good question. Again, something Kristin and I talk about in our course. I love block randomization because you don't always see the term there. It is saying that if I were to just flip a coin for each person coming in, if I had two arms, I don't want to just flip a coin because by chance, Kristin and I keep talking about things happening by chance, I might end up with everyone just getting heads and everyone in the hookworm group, right? And we don't want that. We want to balance it out. So you use a computer to randomize blocks of 6 or 10 or 16 at a time. And that way, you know that within that block, you will have equal number in the treatment and the control group.
Mm-hmm. And then what is the formula that's used to calculate sample size for RCT, and how do we determine primary and secondary outcomes?
So there's formulas that you can use. A lot of times if you work with a statistician, they'll actually do a simulation rather than relying on a formula. So but there are ways to calculate that ahead of time. Primary and secondary really just depends on what is the variable you think is most important, and designating that as primary controls the type 1 error rate on that variable.
Hey, Kristin, you talk a lot about that in our course as well. What makes a good primary outcome based on the measures that you have available and what is valid and what actually is close enough to the sorts of things you want to see? Yeah.
Yeah, we do have a long discussion of that because you need to make sure it's something measurable, something that's going to change, right? Something that's going to be impacted by the intervention and lots of considerations. Yep.
And then another question was, is phase 1 the same thing as a pilot study? Is there
also talked about in the course, these are loosey-goosey definitions. So Kristin and I, statisticians, we like things to be defined and then they mean something. Once we started, looking, it means different things to different people. If you are, sometimes the FDA has very particular definitions, so you can go to fda.gov and find out exactly how they're defining phase 1, phase 2, phase 3. But things like pilot or exploratory study or early stage, those sorts of things in the non-FDA regulated sphere are thrown around a little bit.
Okay, and then regarding case study number 3. Was there any way in this gamification study to control for cheating? So how do we know that the watch was not put on a spouse or a child, for instance?
That's an interesting one. They'd have to be really engaged to care that much. I didn't see anything in the protocol that talked about that. So I imagine they were doing it on good faith. But you're right, they're definitely with the smartwatches as ways to cheat. So if they had been super motivated to beat, Yale Psychiatry or whatever it might have been, I suppose they could have cheated. And because it's measured in with a smartwatch, it'd probably be hard to tell. But I didn't see anything in the protocol about that.
And then another question is, with such flaws such as cherry picking, how do these studies get published?
Regina, you want to take that one?
Oh my goodness, how do they get published? Some of them, not all of them get published, right? So we should preface it that it's not all doom and gloom. There is good research that is being published, but you're right, there is a lot of cherry-picked data out there that is being published. Part of the problem is that I think the incentive structure, right, you need to publish a lot. There are people who really need to publish, and also the peer review system. So Kristin and I do statistical peer reviewing, but typical peer reviewers are interested, they understand about the content, the medicine of whatever it is, and they're not looking at the methodology necessarily. So it's very easy for these sorts of p-hacking things to slide underneath the radar. And if you're very determined, you can hide your p-hacking from peer reviewers. And that's why we've seen a lot of scandals come out recently with researchers who have been caught doing fraudulent things with data.
Yeah, peer reviewers, I mean, they're overworked. It's volunteer work. So sometimes the quality control is not as good as it could be.
Okay. And then another question here, based on case study number 3, was how should we deal with the drop-off in dedication or participation that we often see in long-term studies?
So another thing we talk about in our course is participant engagement. How do you keep participants engaged? Because if you have nobody using the app at the end, that really does water down your results. So there's definitely things that you can plan to try to keep those participants engaged, and super important because you don't want a lot of missing data.
So I know we're coming to the end here. We've gone over a little bit. So just a couple more questions if we have time about case study number 4 here. We had, like I said, a lot of Q&A questions come in. And so the next question is regarding case study 4, what's the difference between micro-randomization and crossover design?
Yeah, good point. I tried to address that a little bit along the way. So the micro-randomization is very cool. It's a very new technique. As Kristin demonstrated, you can't do that by hand. If you are going to randomize everyone to a new either treatment or control group every day or every week, that is not something you do by hand. So the crossover design typically is used to refer only to you crossover one time. So usually it's something if there are only 2 arms, maybe a treatment and control, or at most, maybe 3 arms, something like that. So you're crossing over, but you're not during the study being randomized to a different condition every week or every day. So the micro-randomization ends up being a little bit more difficult to analyze. There are good statistical techniques for that, just because it's a little bit more complicated, but it's also a little bit more powerful. I hope that helped.
We called a special case of a crossover study, I guess.
Right? Yeah, that's better. That's better. Thank you.
Okay, and last question. The last question here is, what is the most simple way to differentiate primary objectives versus secondary objectives?
So I think that's a little bit similar to the question about primary and secondary outcomes. So is there a more interesting question right after that?
There's a question about how to calculate sample size for crossover studies, and if you can give more information about that.
Yeah, that will refer you to references, so we won't go through that on the board here.
Work with a good statistician.
And then, do you think it's necessary for journals to amend their guidelines regarding clinical trials to mandate pre-registration?
Oh, good.
A lot of them already do. And I think this is why, as we were talking about, you might see people registering right before they publish their paper, because it's a requirement of many, many journals that you are registered, but they may not be checking carefully to see if it was actually pre-registration. They're just looking that you have that number. So if a journal editor or reviewer is checking carefully, they should make sure it's pre-registration. But yes, a lot of journals do have that requirement.
I've been involved in some groups, and this is actually a big discussion among researchers and publishers, because some researchers don't like to be tied down to pre-registering things. They want the flexibility to analyze things as they come in and have some fun. And so they are resisting that, and publishers want it, there's that normal tension there. So stay tuned. That has not yet been resolved.
Thank you. Okay, I think that's about all of the questions that we have time for. So thank you for staying a little bit over here to answer some of these Q&A questions. And, if you'd like to learn more from Dr. Sainani and Dr. Nuzzo, we encourage you to join our clinical trials course. So you'll have more than 12 hours of content to learn from them, access to videos and exercises to sharpen your skills. We also invite you to check out more courses from our health innovation topics. So we have a course in digital health product development as well with Dr. Bryant Lin and Dr. Oliver Alami, and other courses from the Stanford Center for Health Education. So feel free to visit our site. We'll also be sending out a link to the recorded webinar within a week. And so feel free to revisit this content as well. In the meantime, we hope you have a great day, a great week, and we hope to see you on future sessions. Thanks so much.
Thanks for coming, everyone.
Thanks, everyone.